Alright guys, so I couldn't be there today, so I wanted to go ahead and go over some of the words with you via video. I did go ahead and type them up so you can get them down into your notebooks. I will be looking for that eventually, so make sure that you copy down the words in your notebooks. And I did type them up, but I'm going to go ahead and go over them orally like if you were watching me through a meet. So the first word is clone. What is a clone? A clone is a same exact genetic copy. Okay, it has the same exact genetic information as the original. That's what makes it a clone. All right, again, same exact genetic information as the original. It's a copy. You've probably seen it in Star Wars, Clone Wars, you know, the clones. So all the dinosaurs from Jurassic Park, they were clones. So go ahead and write down that word. First word is clone. Alright, so the second word is genome. Genome is all, think of it as all the letters that make up the human gene. The human genome has about 3 billion base pairs. So think about 3 billion connected letters. A, T, C, G, remember that? 3 billion different pairs of those. So that's a lot of letters. In the movie Jurassic Park, they show you how that would take a long time for you to look at it. I'll show you a clip. Check out the clip.
These once a second for 8 hours a day. It'd take 2 years to look at the entire DNA strand. It's that long.
Yeah, so that's the human genome. It's all the letters, all the base pairs, 3 billion in humans. So all letters that make up your genetic information. Alright, so the next word is genetically modified organisms, or GMO for short. That's any organism. Organism is a living thing, remember that. That has had its genes, its letters, modified or changed by humans. Humans have taken the genes of corn, for example, and they have modified it, they have changed it so that pests, you know, things that we usually eat, the corn, would not want to eat it anymore. So become like more resistant to pests. That would be an example of a genetically modified organism, the corn. So anything that has been changed by humans is considered genetically modified. I got a video that I'll show you later so you can look at it, and it really goes into genetically modified organisms. It's pretty good. Alright, restriction enzymes. There are enzymes used to cut or fragment the DNA into different sections. Again, restriction enzymes. Enzyme that is used to cut or fragment the DNA into different sections. Alright, so the next word is genetic engineering or transgenic manipulation. Okay, genetic engineering or transgenic manipulation. That's the next word. And what that is you're going to change or manipulate the genes of an organism. Like when you take the genes of a glow bug and you put it into a fish, and now that fish glows. Okay, that would be considered genetic engineering or transgenic manipulation. Transgenic means you're transferring, transgenic, I'm transferring the gene from one to another. So that would be transgenic manipulation. Like I said, like when you put the gene of the glow bug into a fish and now the fish glows. They've even done it with mammals too, where they can make mammals glow. You know, hey, maybe in the future we can glow. They could put the glow gene in us and you can go to the clubs, you know, and we'll be in the club dancing and be like, I'm glowing. You know, I think that'd be pretty cool. Again, transgenic organism is the next word. Transgenic organism, I've kind of talked about it with the previous word, right? Transgenic, again, you're transferring the genes. Organism is anything that's living, so transgenic organism. So basically put an organism that's had its genetic structure changed. If it's had its genetic structure change, when I said, like with the glowfish, for example, again, that would be a transgenic organism. Now the next word is transgenic bacteria. So why do I go into this transgenic bacteria? Maybe you're thinking, Okay, I get it. Transgenic, you transfer the genes from one thing to the other. So why are you going to say it about bacteria? Because on the test, they like to talk about transgenic bacteria. So basically, this is a bacteria that's had its genes altered, okay, by receiving transferred human genes, for example. You transfer the human genes into the bacteria. Like you transfer the human gene that makes insulin into the bacteria, and now the bacteria is making human insulin. And being that bacteria reproduce asexually, make copies of themselves a lot, then they will continue passing on that gene that was transferred, that human gene that was transferred into the bacteria. They will keep on reproducing it and creating more insulin. And now you can give insulin to people. It's a lot more cost effective and a lot
for your
better.
rest of your life. We used to use insulin from pigs, but it doesn't work as well as human insulin. Now the problem is, how do you make enough human insulin for millions of diabetics? The answer is to get the gene that makes human insulin from a person's DNA, cut it out and stick it into a bacteria. The bacteria will read that gene as if it's one of its own and manufacture the protein insulin as if it was one of its own proteins. So you're basically using this bacteria a bit like a slave to produce human insulin. The great thing is that once you've done it once, the bacteria will reproduce millions of times, pass on that gene every time because it reproduces asexually, and so very quickly you can get a population of billions of bacteria all making human insulin in a little factory for us via genetic modification. So to go through that process in detail, step 1, remove a plasmid from a bacteria. Cut it open with some restriction enzymes. Step 3, locate and remove human insulin gene using the same restriction enzymes. So you're going to cut out the human insulin gene from someone's DNA using those same restriction enzymes. Then you need to combine that insulin DNA with the plasmid. To do that, you're going to need your ligase enzymes, so then they get combined. Step 5, the bacteria are then incubated with the new recombinant plasmid and they take it up. The bacteria will then take up that plasmid, as I said, and start to produce the insulin. You can then get that bacteria to reproduce and it will pass that gene on to all the other bacteria.
All right, this next one's pretty easy. It's DNA fingerprinting. Fingerprinting is used to know each person has their own individual fingerprint, makes it unique. DNA fingerprinting is basically everybody has their own unique DNA. So you could look at the person's DNA and figure out who they are. You can identify them through their DNA. So that's called DNA fingerprinting. And just like in fingerprints, you would look at like the lines and the curves and all that. In DNA fingerprinting, you would look at the repeat patterns to see if they are similar. All right, so the more similar they look, the more likely that they're related, or if it's very similar, then it's the same person. So DNA fingerprinting, think of it like regular fingerprinting, except you're using DNA to figure out the identity of the person. So stem cells can change into any cell in the body. Stem cells are amazing because they, how you have like brain cells and you have skin cells and all that, muscle cells. Well, stem cells, they can turn into any of those. So they're kind of like, remember when we talked about the meristem, in the plant, they can turn into any part of the plant. Okay, think of stem cells like that, but for humans. So imagine someone has brain damage, they could actually get the stem cells, and that might repair their brain damage. Or someone is paralyzed, and the stem cells might be able to fix the nerves to not make them paralyzed no more. So stem cells are pretty neat, and there's a video I want to show you about stem cells. Go ahead.
There are these wonderful cells that exist called stem cells. These cells have the ability to become any type of cell because they are not specialized. Scientists were hugely excited by their discovery because they can be used to repair damaged organs, bones, or cartilage. They could also be used to help treat diseases, such as by making new brain cells to treat people with Parkinson's disease. Or they could be used to repair damaged immune systems, and even reverse paralysis or regrow lost limbs. The discovery of stem cells is huge for modern medicine and could have a massive impact. There are two types of stem cells in humans: embryonic stem cells and adult stem cells. Embryonic stem cells are unspecialized cells that can develop into any type of cell. Adult stem cells are also unspecialized cells, but whilst they can develop into many types of cells, unlike embryonic stem cells, they cannot develop into every type of cell. Adult stem cells come from tissues such as bone marrow. As in the name, embryonic stem cells can be removed from unused embryos, such as those left over from fertility treatments, for example.
Alright, so the next one is gene therapy. Gene therapy is basically I'm going to try to fix a defective gene in a person that's giving them a disease. And by doing that, by inserting a gene into them. So I put a gene into you, and then hopefully that will fix your defective gene, and then you'll be okay. So that's gene therapy. It's pretty self-explanatory, right? Gene, and then therapy is you're trying to fix something, right? So it's pretty easy to know that one. All right, and the next word is gel electro. I'm not even going to say the whole word. Just say gel electro. All right, sounds like a villain or something. But basically, it's just it's a machine that's used to separate the DNA. That's all you got to know. It's a machine that's used to separate the DNA.